फ्री हो जाएगा ना। A very good afternoon to one and all. Before proceeding, may I request everyone to please switch their phones on silent mode. So, I am Chaitali Singhal, and it's a privilege to welcome you all to the 5th Professor M.K. Bhan Memorial Oration. Today, we gather not just for a formal event, but to honor the life and work of a visionary who redefined science and public health, especially in the Indian context. Dr. Maharaj Kishan Bhan was more than a brilliant physician scientist. He was a relentless advocate for children's health and a transformative force in translational health. His journey began at the Armed Forces Medical College in Pune, where he developed his unwavering commitment to medicine and service. This dedication evolved at AIIMS, where he transitioned from a clinician to pioneering researcher. Driven by a vision to bridge science and real-world impact, Dr. Bhan's work not only advanced pediatric health, but also set new standards in equitable healthcare solutions. His legacy is a reminder of the power of science to create lasting change, especially for those most vulnerable. Dr. Bhan's legacy is deeply woven into real-world solutions such as the integration of ORS and zinc into national health policies, which continue to improve countless lives. Among his stellar contributions is the development of the rotavirus vaccine. From identifying the strain in the neonatal unit at AIIMS to overseeing its development and its eventual inclusion in national immunization programs. His vision extended far beyond medical science. This is evidenced by his establishment of 8 diverse institutions under the Department of Biotechnology, which focus on various disciplines beyond healthcare, including agriculture, genomics, and animal biotechnology. As Secretary of the Department of Biotechnology, he brought the same compassion and vision to an even broader stage. He championed transparency, inclusivity, and interdisciplinary collaborations, thus reshaping the current scientific landscape. His work with vaccines and his vision for a more accessible scientific framework set the foundation for growth in this field. Today, as we sit in the M.K. Bhan Auditorium at THSTI within the NCR Biotech cluster, a cluster that was his brainchild, we are reminded of the passion, dedication, and wisdom to shaping a healthier, more equitable society. This oration is more than a tribute. It's a call to continue his vision of science serving all. It's a reminder that each one of us has a role to play on building on his legacy, in making science work for the good for the society. With that spirit, let's begin today's program. I would now like to invite Dr. G. Karthikeyan, Executive Director, THSTI, to open today's event.
Good afternoon. I don't know why they gave me that. That was unexpected. So it's good to see this auditorium full again. So good afternoon, everyone. And I welcome you all formally to this 5th Dr. M.K. Bhan Memorial Oration. As you see, this is jointly hosted by THSTI and RCB. Dr. Arvind is also here. And I'm sure it's not lost on everybody that Dr. Salunke is giving this oration today because he's overseen the development of both THSTI and RCB. So In that sense, it's a coming together of sorts. So welcome, sir. Dr. Jayanta will be introducing the guest formally. I only have this to say. The topic that Dr. Salunke will be speaking on, it signifies what THSTI is going through this time. Oh, I'm sorry. I think Dr. Shinjini will be introducing, so it's okay. That was my fault. Sorry about that. No, so what I was saying was that as THSTI is now becoming a full-fledged translational institute, I think the topic of your talk, sir, is very appropriate, and hopefully it will ignite minds here, not just the faculty, the younger faculty, but also the students here. Thank you, sir.
Thank you, sir, for your kind words. Further, I would request Dr. Arvind Sahu, Executive Director at RCB, to speak a few words in the memory of Dr. Bhan.
Good afternoon, everyone. On behalf of RCB and on my personal behalf, I welcome you all to this oration, special oration in the memory of Professor Bhan. It's good to see everyone together here. It's like a larger family coming together. And particularly grateful to Professor Salunke, who is part of the family actually, who, you know, along with Professor Bhan, initiated this whole thing during discussions and everything. So thank you so much, Professor Salunke, for accepting the invitation. It's pretty much organized everything by actually THSTI, and I just piggyback on them to add the RCB name. So it's pretty apt, like, you know, Dr. Karthik said that his work and THSTI work is quite complementary. Professor Salunke, of course, everyone knows him, the entire scientific fraternity of India knows him and his work, particularly on the immune recognition and at the molecular level, and how the antibodies really recognize antigen, and is particularly about the specificity. Is it really specific about what we say, or can they recognize a little bit structure and then there is some play there? And, you know, how did he come up with this idea is quite exciting, and we'll hear today about his work and, you know, at the molecular level, how the whole thing really works. Quite interesting and quite fascinating and unique idea which he developed, you know, which till the work came, people were very kind of bogged in this idea of specificity, that has to be like this, has to be like this, and doesn't work the otherwise. As we all know, Professor Bhan was a truly visionary leader. For me, almost, you know, everyone who is in the gathering knows such a visionary person who, you know, not only contributed to various DBT institutions, thought about this cluster idea, that how do you bring in together the different kind of institutions, and, you know, also created many of the things, including the India Alliance, you know, how to get the people from outside, put them in different institutions, not just DBT institution, but create a mechanism where, you know, people who are doing so well can be brought back and placed in them, which has done so many, many things, you know, truly, which he has started in DBT, but not just for DBT, for the entire Indian science. And that's something, you know, a visionary leader always do, is that not think, you know, in that let's think only about our own comfort zone and our own institute or our own department, but think, larger picture. Of course, his work everyone knows on rotavirus and what he did, so I'll not go and get into things. I'll tell you personal incidents with Professor Bhan. The first time I met Professor Bhan was in NCCS, where I was at that time junior faculty working thing, and Dr. Mishra, who was a director at that time, always had this habit of anybody comes, he'll always praise his scientist and say, "Oh, dekho kitna acha kaam kar raha hai." This, and Professor Bhan and him, they were walking actually, and he was in hurry because he had to catch a flight. And so they were just passed by, and I said, "Hello, sir." And he said, "Is Arvind is doing wonderful well. Abhi iska JVI mein paper aa gaya." He was actually gone. He turned around and came back and congratulated me, right? So this is a person, he was a people person. He knew how to encourage youngsters. It didn't matter to him, one small junior faculty in an institute, how does it really matter to him? Is he could have said, smiled and just walked, but then he turned around, came and congratulated. And that's something, you visionary, someone who is this kind of person, a people person, having empathy, can only do that, and that goes because, this memory stayed with me and I still remember. And and this was not a person specific. He would encourage almost everyone, also get the right people, encourage them, bring them back, right? So we know how he got people and got the work done. So Professor Bhan's vision was to create really a vibrant, biotech cluster, which will have different kind of program, different sciences. And I would like to, inform all the people who are not aware that this is pretty much in the agenda of DBT right now, and they are thinking, and we are discussing. There are multiple meetings that happen, and I don't know if Manish Diwan is somewhere here, but he is working and looking into this larger infrastructure of converting this biocluster into a biopark where multiple institutions with the specific domains come in, talk to each other. Multiple facilities will be created, many more startups, pilot plants in various technologies, also the industries, everything which come together, and this becomes a vibrant. So DBT is pretty much going in this direction, and a lot of thought is given. And a person has been now appointed to look into this, to make this bigger, more roadmap and discuss with everyone who are partners in this. So regarding the programs, I would like to tell some of the people who are not aware. The DBT is also thinking in terms of the larger, not just this cluster, but also virtual cluster, bring in the other institutions together, bring in bigger program. And RCB's mandate, of course, is research-based learning. And towards this, bringing program, we have started I3C, which is conceived by DBT and given the responsibility to RCB to steer this program, where all the 15 DBT institutions are, have come up together to make this and part of this program, where the students, are recruited. They're recruited in a network project where PIs from multiple institutions are part of a project, and these the students are part of that. They are, right now with us for 6 months of training at RCB, where they will be learning not what they have learned, but something which they are not exposed to, right? And also every single day experience in the high-end platforms which they will go, followed by the immersion program to really learn the ground realities of India. And that's something they are going to do. And then they will be going to, so a lot of these kind of things, clusters and also bigger things are there in the pipeline of DBT. This, of course, for a cluster to happen and get bigger, will also have its own challenges. The biggest challenge I see is the equity, because when you have multiple stakeholders coming together, how do you do that? How do you bring everyone? How do you steer the program? How do you create but there was some idea already floated by Professor Bhan, which was there of creating an office of connectivity so that the whole thing works in a smooth way. Maybe that's the way to go. So he was not just thinking about the cluster, but also thinking about the growth of the cluster and then the administrative structure of the cluster and how do you steer. So great visionary. Thank you so much, everyone, in particular Professor Salunke, for coming, and let's hear it. Thank you so much. Thank
you. I would like to request Dr. Karthikeyan and Dr. Sahu to please be seated on the stage. So yeah, moving ahead, it's my pleasure to welcome Professor Shinjini Bhatnagar, Distinguished Professor at THSTI, to introduce our distinguished speaker and chief guest, Dr. Dinakar M. Salunke, former Director, THSTI, RCB and ICGEB.
Good afternoon, everyone. I think a lot has been said about what Dr. Bhan thought for us. His firm belief was that fundamental science, bridged with innovative clinical and translational research, augmented with deep education, is the best way of addressing the country's needs and the health challenges. And that was the vision with which this whole cluster was conceptualized. Visions often remain dreams, but here we witnessed it actually being implemented. And therefore, it's an absolute honor to welcome, on behalf of the NCR Biotech Science Cluster, Dr. Dinakar Salunke, who's been a foundational pillar in actually the application of this concept and vision. Dr. Salunke has, as we all know, has had a very illustrious career. He was awarded a PhD in molecular biophysics from a very prestigious Indian Institute of Science, Bangalore, in 1983, and then went on to do his postdoc training in Bandia Institute, Waltham, Massachusetts, after which he came back to India to pursue his interests in structural biology and immunology. He established and led the group of structural biology at the National Institute of Immunology for almost 25 years. In 2009, he took up the challenging task of becoming the founding director of the Regional Center for Biotechnology, which was truly being an outlier at that time. It was a challenging role. After contributing immensely to the institute and to the cluster, he transitioned to another leadership position at the International Center for Genetic Engineering and Biotechnology, where he served for 7 years. He is currently an emeritus scientist at the same institute from January 2023. Dr. Sahu has already talked about, and Dr. Karthikeyan, about his work on structural biology and immunology, and we are going to hear his exciting contribution very soon. I just want to add that he was one person who also saw that science alone, without the application of clinical and translational research, will not lead to an application that is important for solving a clinical health problem or a public health problem. And his contribution to the maternal child health program at THSTI was very, very encouraging. And that was truly a multi-institutional program on the cluster, something that was envisaged for this cluster. So I think for us personally, Dr. Salunke has played a major role in the building of the two institutes and the cluster, right from the interim facilities at Gurugram to coming to this and building this expansive cluster. He has been a constant guiding force to be on the path that this cluster was envisaged for. So, absolute pleasure and honor to have you here, Dr. Salunke, with us. Please.
Good afternoon to all of you, and thank you, Dr. Karthikeyan. Thank you, Dr. Sahu. Thank you, Sinjini, for those nice words. I don't know how much I deserve that much of a praise, but I'm indeed honored to be here today to be a speaker for the M.K. Bhan Memorial Oration, considering that I hold Professor Bhan at great esteem. I actually do not know Dr. Bhan for a very long time. In fact, my interaction started with Dr. Bhan since 2005, when I was given charge of National Institute of Immunology as acting director after Dr. Sandeep Basu retired. And during those 3 months, I had a fantastic relationship with Dr. Bhan, and that's when I discovered him rather. Probably he discovered me also. And then subsequently, after about a couple of years, I was being interviewed for a position of deputy director in the National Institute of Immunology. And Dr. Bhan, as expected, was chairing this meeting. And interview went very well. And by the time I was about to walk out of the boardroom in NII, Dr. Bhan, it appeared to be joking with me. And he said, look, you will have to take additional responsibility, not only be deputy director, but as a within NII, but beyond NII. And he told me that he will sign the minutes of that meeting only if I agree to take charge of the extended Faridabad campus, which was to be RCB and THSTI. And I actually thought he was joking, and I said, yes, no problem. And that is when I didn't realize that I was really making a big commitment. I got the appointment as a deputy director in NII, but within about a week, I also got additional charge of these two institutions. And we started in basement of what was designed to be faculty lounge at that time, in NI campus, basement area was sort of given to us, and then now that actually that building in NI is a library, because they moved the library outside the main building, and we rented out couple of buildings for THSTI and RCB in Udyog Vihar, and we started operations in variety of directions. Luckily, we were able to recruit Sri Kumar Suryanarayan, who as a CEO, and he's one of the main responsibilities was to take care of activities here. And that was great relief. Sri Kumar actually began this extremely well, and we were operating in Gurgaon, Udyog Vihar, Gurgaon. And then one day, I'm telling you an anecdote which was very turning point for me. One day, early in the morning at 6 o'clock, I was to take a flight to Bangalore, and I was about to sit in the car, and I get a phone call from Dr. Bhan. And my flight was very early, and I was already getting late because the driver had come little late, and I was tensed up. And I get this call from Dr. Bhan, and he says, what is the status of the tender for the project management company appointment? I said, it is being handled by Sri Kumar, and I'm sure he would be taking care of it. He got even more upset. He said, what do you mean by that? You are executive director and you ought to know what is the status. I realized that I have a job to do. So after the call from Dr. Bhan, I called up Sri Kumar, and he told me that the advertisement for the tender has come a couple of days back, so everything was in order. And I called back Dr. Bhan and told him. So obviously, Dr. Bhan seems to have been sort of misinformed of the situation. But one lesson that I learned from this is that I'm supposed to know everything all the time. And sure enough, because Sri Kumar did not stay for too long, he, within a year of doing initial work here, Sri Kumar went back to Chennai, and then I think Bangalore later, but to set up a C6 C6 energy company, and the entire action actually took basically fell upon me. And then that lesson that Dr. Bhan had told me I have to know already, that became realization that if I didn't know anything, I would have been blank when Sri Kumar was leaving. So of course, all this what happened was not done by me alone, both by my own efforts as well as efforts of Dr. Bhan. We had an excellent team, particularly fantastic effort, fantastic support from officers in DBT, Dr. Sinha and Dr. T.S. Rao. Dr. T.S. Rao happens to be here today. And the finance advisor, all of them were fantastic supporters of this cause. And also we had Sudhanshu Vratti here, and many other people we had put together. And this program started moving forward very smoothly. We also had a support from Martha Gray from MIT, and what Dr. Bhan had conceived, as it has been pointed out by everybody, was actually put together in a slide in 2009 collectively by contribution to all of us, and that is, where is the slide? This slide actually tells everything what Dr. Bhan conceived, and we reflected it on the paper to be what this cluster is. And I see it happening in that direction more or less. So RCB was meant to be an academic institution. And remember, I'm talking about 2009, 15 years back. THSTI was meant to be translational. RCB was to take care of research and education and training. THSTI to be research, development and business, and new center of clinical health research, which was to be developed yet. We didn't start it then, but I saw in the afternoon today a building that is coming up for the clinical research. So with that being there, we have technology platforms in ATPC, we have animal facilities right in front of this complex, and all the program seems to be moving in right direction. And this vision of Dr. Bhan implementing, I'm very proud to have been part of it. And so many of the people, some of them are still here, some of them are not there. One good person name I actually ought to be remembering. Immediately in 2011, a couple of years later, Dr. GB Nair joined us to be executive director of THSTI. And we had a tremendous wavelength match. We together worked in such a way that we almost had absolutely no difference on anything. And this moved forward to the extent that in a record time, the first phase of this campus was completed, and without any audit paragraphs of any kind of significance. So I think it has a clean record, considering that there were many institutions which were established around that time all over the country, and they would have taken this 3-year time that we spent has been 10 years for some institutions, which some of them have actually bogged down into legal problems. We managed to solve those problems very neatly, nicely, with very innovative ideas. And I will not go into the details of those because that is not main part of it. But since Shinjini suggested to me that I should tell little bit about how the cluster was established, I thought this would be the best slide. I spent about 3 hours finding this slide in my old emails, and I was able to get it. Where is the pointer? So coming back to the topic of my presentation, actually Arvind almost stole my thunder by preempting what I'm going to speak. So I really have to do something beyond that. As he mentioned, so that also would help me to reduce the time that I spend on my slides. You know, as early as almost 90 years ago, Landsteiner has done a very elegant experiment in, actually, he was really a chemist, very good chemist, and he showed very small variations in a small molecule, which is aminobenzene, and variants of it. You can see if you generate immune response against one of them, it actually shows no cross-reactivity of others, which are only one functional group different between each other. And that laid down a situation with regard to flexibility, a very simple chemical experiment using anti-sera from the mouse model. And about 50 years later, this was illustrated crystallographically. You can see a antibody against lysozyme. This is the antibody and this is the antigen lysozyme. You can see by pulling out these two molecules, the great complementarity between these two. And that complementarity is practically like a lock and key. And that lock and key model is something that everybody believed for long, long time. And there are thousands of structures of antigen antibody interaction have been determined. And specifically, to cut the long story short, the discrimination between self and non-self was really put together in terms of physicochemical principles of molecular interactions. So physiological process of non-self recognition was put together, and how the identification and exclusion of foreign antigen would work is essentially large number of antibodies to the epitopes on the surface of the antigen will bind and sort of neutralize it. This is something which we knew and we all understood it. So in that sense, there was nothing more to be done about this in the particularly in crystallography, because I'm saying because I came to Immunology Institute and I was supposed to work on immunological problems using my skills in structural biology. So what next? But one thing one has to remember is that host pathogen interaction, the immune response is what the response from the host is, and pathogen is what the foreign antigen would be, is not something like this, which is a rock being a rock-like structure with a definitive three-dimensional entity is being recognized by the antibodies. But it has complexities associated with it. And those complexities, if you look at it, you will appreciate what I'm saying is right. Simplistically, this all seems to be complete and perfect. But when you see the context, you will realize there are issues. So there is a dynamics and diversity associated with host pathogen interaction. And number one, with regard to that, we have done quite a lot of work there, but one recognizes the number of antigens, and each antigen is not being recognized by one antibody. Large number of antibodies will actually be required to neutralize that antigen, and millions of independent non-self components are there. In large number of, in fact, the microbiome is huge. And even you don't have to have microbiome, even a chemical entity like aminobenzene, it actually can generate the immune response. And you need huge set of diversities, means that many more antibodies. So number of antibodies that you have to generate, that means you have to have genes to code for them will be infinite. And that becomes something more complex than what I showed you in terms of recognition specificity. The other thing is, we know pathogens are not dead entities, they're intelligent living forms. So they can change their identity over a period of time simply to deceive the immune system. Simply speaking, in terms of antigen, protein molecules can change their structure, or even smaller molecules can change their structure. So what happens if antigen changes shape? The antibodies that are generated against that antigen will become redundant. And that is another question that goes beyond the conceptualization of structural basis of specificity. And third is, again, related to the intelligence of the pathogen. You have discrimination between the antigens. And one thing is foreign entity can actually take a shape of a self molecule, and that will lead to sort of cross-reacting antibodies and so on. So we have actually worked on all of these things, and I'm going to show you a couple of slides in each case and then move on. So first is millions of independent non-self components are to be recognized. How do this is essentially primary antibodies or germline antibodies or receptors on the B cell will be recognizing this infinite pool of molecules and with high level of specificity of the type I showed in that figure. And solution to that problem was actually provided. I didn't have to worry about it. Several years ago, Tonegawa got Nobel Prize for actually coming up with a genetic solution to this in terms of VDJ recombination. So number of different specificity antibodies can be generated very fast by recombination. However, there is a discrepancy. Even if genetic recombination can work, the number of possible recombinations will lead to about 10 raised to power 8 antibody molecules with that many number of specificities. But the number of antigens that need to be recognized is practically infinite. So there has to be something more to it than only having genetic recombination. What is the solution? So one possible solution is maybe antibody recognizing instead of only one antigen, one antibody can recognize multiple antigen. And if that is possible, one can actually think of narrowing this down. And to address this, we actually did experiments with germline antibodies, 2 germline antibodies. And these antibodies, we demonstrated that they do have interaction with large number of. So germline antibodies are actually having multiple molecule binding abilities, and through somatic mutations, they evolve further. So that we demonstrated very nicely in terms of 3-dimensional structure of the germline antibody BB6 bound to 4 different antigens, and the 4 antigens have different structures, and the interacting site on the same germline antibody actually is very different in shape. And this cartoon will illustrate to you what I'm actually showing in crystallographic data here. So different conformational states of the germline. So just like antigen can change shapes, antibody also can change shape. And same antibody BB612H3 can change shape and recognize different antigens. So that generates one level further over genetic recombination. Another antibody 3665, we did 3 crystal structures bound to 3 different molecules. Here the antibody has no change in structure, same structure, but 3 different molecules are being recognized. And if you look at it in cartoon, which is very easy to see, the antibody has identical structure in all the cases, but antigen binds differentially in slightly different places within the antigen combining site. So if I put it together, story goes like this. This is Tonegawa's genetic recombination. You do have different genetic variants of the antibody gene. That is first level of diversity. Second level is actually looking at the conformational flexibility, and that is very easy to understand. But third also is very easy to understand in the sense, simply in that site, you can have juxtaposition of the antigen antibody at a different level. So in other words, level 1, level 2, level 3 together, it actually generates very large number of possible specificities for primary antibody or a germline antibody to be able to recognize large number of antigens. And that is how we were able to give solution to the generation of antibody diversity to be enhanced at 3 different levels. Then the second example that second story that I was referring to is what if an antigen changes shape. So we took antigen, which was a peptide molecule, and this peptide was recognized or identified by NMR and CD data to be a flexible molecule. It was actually identified in a different context. I will tell you that later. But what matters is we know this is flexible. So we got a large number of antibodies were generated against this molecule, which recognized the same epitope in this DPAF. And we thought if we look at the structure of this peptide bound to these 3 different antibodies, we chose these 3 because they were very convenient to get crystals of. And crystallographic studies, actually these crystallographic studies were carried out of Deepak Nair, who I'm sure is here in the audience. What demonstrated, Deepak's studies demonstrated was when there is no antigen, the antibody molecule shows very high level of flexibility. In fact, third one probably is disordered, but these 2, which we have crystal structures of, it shows significant differences in the antigen combining site. But when antigen is bound, you see all of them converge into common structure. So what I was trying to tell you, these 3 are distinct, at least about 6 to 8 amino acids are different in antigen combining site. So although their structures are different in absence of the antigen, they converge into same structure in the sense that in terms of being able to economically utilize the conformational repertoire, there is a strategy that you push the flexible molecule into same structure for antibodies to recognize, thereby actually essentially minimize the cross reactivity. The third point that I was talking about, exploring essentially the cheating that pathogen can do with regard to the immune system, is how the antigenic discrimination can take place. For that, we found almost serendipitously, I won't give you the details of how we landed up with this, 2 completely unrelated molecules. And when we immunized mice with this, one of these molecules, we got immune response that was cross-reacting with other molecules. So what we got is cross-reacting antibodies between a sugar molecule and a peptide molecule. So ability to discriminate between 2 different antigens or not is actually to be evaluated based on this system is what we decided. And when you look at it at the level of polyclonal antibodies, you will not go beyond being able to see just cross-reactivity in ELISA. But if you make monoclonal antibodies, you can actually see the situation in individual clone level. And what actually we saw when we did hybridomas and looked at about a dozen of those antibodies, it was in terms of cross-reactivity, it was very clear about half of those antibodies were very specific. These are sugar-specific antibodies. The other half were cross-reacting. And that was actually a stunning experiment. it was very clear even when we do affinity measurements that sugar and peptide bound in case of this 2D is with similar affinity, and in this case affinity of one bound and other didn't bind at all. So what is clear is that this pool is cross-reacting and that pool is not cross-reacting. So we wanted to actually check if you have antibodies that are the antigen binding to a common antibody, which are probably will have a similar topology. In the sense, if you take since we have talked about flexibility, we have talked about anything else, finally recognition is taking place, as I showed in the first two slides, at the level of physicochemical interactions. So if you have two different molecules, it is entirely possible that three-dimensional topology of those two molecules could be very similar, although overall chemical structure is different. After all, the interaction between antigen and antibody can happen in terms of functional groups in chemistry. And so that hypothesis, when we published this data, was put together, and actually referees accepted that. So we were bent upon demonstrating that these two molecules could indeed be similar. But you can see this is a peptide bound to that antibody, and this is a sugar bound to that antibody, and they don't have any topological relationship. They bind to the antibody, which is not in different conformation. They bind at an overlapping site. So there is no conformational change. There is no differential epitope. It is simply binding through different set of interaction. And if you look at it in terms as a structural biologist, I see no contradiction in all of them. What it means is at the same site, two completely unrelated molecules can bind without breaking in laws of physics and chemistry. So what it means is antibodies actually demonstrate promiscuity, which is something that Arvind highlighted, and that is what we could show here. And then there are at least a couple of examples have come later. One in case of an antibody against dengue surface antigen, which cross-reacted with Zika virus surface antigen, and antibody against one hormone cross-reacted other hormone, and these two molecules are not homologous or similar. So are these two. So these have come after we actually discovered that antibodies do have a cross-reactive ability, and that hybridoma data that we had, at least half of them were specific and other half were not. Actually, the one example which was well known, this is anti-idiotypic antibodies. Anti-idiotypic antibodies actually show antigen and antibody are actually binding to the same anti-idiotypic antibody, but they have surface features different. So the antibody promiscuity is actually a real thing, is what became obvious. And so what we think the summary of this is set of clones which have acquired ability to discriminate. That is actually essentially defining specificity and fidelity of immune response. And another set of antibody clones which show plasticity, which is probably designed to disregard antigenic variations. In other words, if you have a vaccine against virus which has variable surface epitopes, you have antibodies generated which are actually also plastic, and they will ignore those mutations and still bind. So you don't have to worry about influenza and HIV is what it would appear to be. So when we were demonstrating this, we were actually using our system, which was extremely artificial. It had nothing to do with biology in terms of those particular antigens. One is, two are very significantly simple chemical entities, but immune response is only our biology. But real pathogens may not be behaving like this. So people used to ask us, you actually have to do these experiments with real pathogens. And one thing is very clear, is while you are showing promiscuity, there is no problem about this. But what about this? Real pathogens are known to escape, particularly in case of influenza virus and HIV, and probably there are similar situations we have to study a great deal in case of SARS-CoV-2. But what is important here is, if you take influenza virus, you have mutations taking place, which will make mutant not cross-react with the antibodies against the wild type. So this and this seems to be contradictory. So rather than answering this question by looking at an artificial example, we decided to take the problem head on and decided to look at the epitopes from the influenza virus, which is known. And surface, among the prominent surface antigens is hemagglutinin. And we decided to look at what is known neutralizing epitope from hemagglutinin, which is a trimer. And this red is a epitope, which we actually synthesized and made large number of antibodies against it using phage display library. And there were 16 phage clones. Out of that, we took 12 high binders and screened all the mutants that we could think of. About 7 of them, in addition to the native, we checked the cross-reactivity. And almost all of them except one actually showed all the antibodies, all 100 antibodies that we looked at actually cross-reacted with equal population. Whereas this one with a mutation of R did not cross-react. And we have done this by purifying these molecules, at least 12 of them, and showing not only cross-reactivity, but made models and demonstrated this story that happens at a population level here is true at structural energy and in terms of free energy of binding, but I will not show you. But what it actually explains is that there are only finite number of mutations that are important in terms of escape in case of influenza virus. Not all mutations are important. And that explains the, despite high rate of mutations in case of influenza virus, the rate of mutations are very high. But the efficacy of epidemics is less than the number of mutations that you take place. So it explains that you do have some critical hotspots that are important in terms of escape, but not every mutation is important. So I'm changing the topic here. At this point of time, a new student of mine came to the lab, and I was actually trying to ask him to pursue this further in terms of mapping structurally all these antibodies. He was not interested. He said, you're talking about the variable part and what that takes place in mutations, and he wants to look at the invariable part and of the influenza virus. So he decided to concentrate on another antigen of M2 ion channel, and which is a M2 ion channel, and he discovered that N-terminal domain of the M2 ion channel is very constant. It doesn't change. It, of course, is not a surface protein, but it is an important molecule, and this region, in over a large number of strains, it doesn't make any mutations at all. So he decided to make antibodies against this part, and he made large number of antibodies. he purified and characterized 4 of them. So AU1, GU1, JU1 and UU1, and you can see affinities. In fact, these two are 10 to the power -7 affinities, whereas this is micromolar affinities. So he took all of these antibodies, expressed, purified, and subjected them to both functional and structural studies. We had a collaborator in JNU who had established this assay of looking at influenza virus propagation. And you see, you have this cell line where you add the virus and virus enters and it replicates and comes out also multiple copies of it. So the virus propagation takes place. When it takes place, you have, if you precipitate and separate the virus cells, and you will see virus in terms of the RT-PCR in this part also and this part also. So the supernatant also and the precipitate also. But what if you put the antibody here? If you put antibody and it is actually effective against this virus, what would happen is you may have virus here, but it may not have virus here. And he did exactly this experiment and demonstrated out of the 4 viruses, in all 4 of them, of course, were found in intracellularly, but one of them, AU1, was absolutely not available in supernatant. Others were at a different levels. So what one can take is AU1 seems to actually inhibit propagation of influenza virus. So he then did a crystal structure of AU1 with the M2 invariant peptide, and he was able to get crystals. This is a dimer of antibodies bound to a dimer of peptide. So it's 1:1 complex of antigen antibody. And it is quite clear that you can see the peptide very unambiguously. It is actually intertwined into a beta sheet kind of structure with hydrogen bonding, which is very strong. So you could actually see relatively low resolution map was showing very distinct even bonding, hydrogen bonding. And if you go back a little bit and look at how M2 is known to function, this is the M2 molecule and this is intracellular part. You can see it exists as a tetramer. So it works as a ion channel only as a tetramer, and this N-terminal part, which is actually constant, comes together. So, so that is incorporated in virus assembly, and functional virus is like this. What we have is a antibody which is binding to this region, and exactly in this is intertwined in the real structure, but this is a schematic. So if these 2 antibody molecules bind, it will lock it into a dimeric form. It will not form a tetramer. If it is not a tetramer, it will not work like an ion channel, and so no functional M2. So it essentially the antibodies made against the M2 invariant peptide seem to be not is not allowing the tetramer formation. So they're not allowing the assembly to take place. If it is not assembled, it will not come as a full virion. And that is actually the story that we discovered. So what it actually conclude, and the 3 other we didn't try, rather we tried, but we were not able to get crystals of that. So AU1 alters the quaternary state of M2 protein and inhibits assembly of viral components in the host cells, and thereby replication, the propagation and release of this is interfered with. I want to tell one more story of antibodies, but I don't know how much time I have. Over. Okay. So this was very designed peptide in terms of making therapeutic translation. So this is very important in terms of translation as much as the previous study. Another student of mine got enthusiastic about making therapeutic antibodies against snake venom, which everybody is talking about them doing it, but we don't see anything happening, at least in the literature. So my student began searching for what has been done and what can be done. And we got from the Irula snake forms, cobra and viper snake venoms, and he made 5 antibodies against cobra directly using snake venom. So screen the library with snake venom and you pick up antibodies without worrying about which toxin it is binding, where it is binding, nothing. And he did a couple of them with the viper, and purified, expressed, and antibodies were available for both characterization. One of the things to do is to find out which antigen it binds to after purifying. Actually, some of them are not very high binders, but I will leave that. What we did is we did mass spectrometric analysis after pull down assay, and we could see 6 antibodies which were 4 of which were binding to phospholipase A2, and 2 of them, one of them is nerve growth factor binding and other one is zinc metalloproteinase. So we took 4 of them, which were all against the PLA2, and proceeded to do animal experiments. And you can see with NASA antibodies, N194 and N248, you can see 50% survival. In fact, it increases life longevity much longer, but for the duration that we looked at least 50% of them were very effective, and 50% of them were surviving, and in this case, one of them survived. Although when we tried to mix together, it did not increase. So there is no benefit of co-administering, but at least one antibody we could get, which was very effective. We did it with Echis carinatus and same thing, but one advantage here is when you add 2 of them, it actually works a lot more effectively. So in other words, some combination here works in a synergistic manner, some work individually. At least one of them works at 50%, and this in combination works at about 33% in terms of improving the survival. So we did experiments to check how does it work in terms of actually showing inhibiting the effect of snake venom, and we did experiments on hemolysis and myotoxicity. I will not go into the details, but I'll tell you briefly what the results are. So myotoxicity was actually affected by all the antibodies at different levels, but only E113 inhibited the venom-induced hemolysis. So we, of course, didn't do neurotoxicity, which is an important experiment to do. Nevertheless, we had actually biochemical analysis to support the animal experiments to illustrate that these antibodies are inhibiting effect of the venom at the biochemical level. And these are most important toxicity, hemolysis and myotoxicity, it actually affects. So in other words, we were able to reduce the toxicity of human components, and oligoconal combination can actually work through. So if we can actually generate a large pool of antibodies, we can really effectively make a possible therapeutic solution to antibody therapy solution for the snake venom biting. I'm not going to go on further. I think I have talked subsequently. I've given a long story which started at NII, and some of the people whom I have highlighted here have contributed more significantly. As I said, Dipti Jain and Deepak Nair, who are here, have hugely contributed to those initial experiments in immunology. And Ujjwal Kumar and Amit Kumar were doing these therapeutic antibodies. Amit Kumar was on snake venom and Ujjwal on the generally mostly my students went in a way in a directions that I initially started because they were adventurous, but they have been very successful. The initial immunology experiments were actually done in collaboration with Kanuri Rao, who used to be here at some stage. But many of my friends have collaborated, but I think the first part of this, I need to acknowledge Kanuri Rao. And the influenza virus studies I had done with Dr. Rajla in JNU. And as I said, NII, RCB, as well as ICGEB have actually supported me a great deal in terms of this science. And thank you for your attention. Oh, no questions, is it? Yeah, I thought.
A very thank you to you, sir, for such a descriptive introduction and the brief. So I now request Dr. Karthikeyan, Dr. Sahu, and Professor Bhatnagar to felicitate Dr. Salunke. Thank you, everyone. So to bring our gathering to a close, I'd like to invite Dr. Arun Gurung, MK Bhan Fellow, for concluding remarks and the vote of thanks.
Good evening, everyone. On behalf of THSTI and RCB, thank you all for joining the 5th MK Bhan Memorial Oration Lecture. This annual event is a celebration of Dr. Bhan's legacy and his commitment to advancing science for society's benefit. Our sincere gratitude to Dr. Dinakar M. Salunke. Sir, your deep insights into antibody biology and their therapeutic design will stimulate others in the field to push existing boundaries towards betterment of science in general and health in particular, both in the very spirit of Dr. Bhan's visionary work. We also thank Professor G. Karthikeyan, Executive Director, THSTI, Professor Arvind K. Sahu, Executive Director, RCB, for their constant support and direction. We also thank all the distinguished dignitaries from DBT, other institutions, and several funding agencies for being able to join us online. Special thanks to Dr. T.S. Rao, who is here with us today. We thank all THSTI and RCB faculty, scientists, colleagues, PhD students, research fellows, and admin staff for their support. Your presence and work reflect the far-reaching impact Professor Bhan has on all inspiring professionals and young scientists alike. I take this opportunity to specifically thank all the committee members, THSTI admin, and IT team for their sincere efforts to ensure that the entire event runs very smoothly. As we carry forward the insights shared today, let us each commit ourselves to the values Dr. M.K. Bhan championed, that is, innovation, inclusion, and the pursuit of knowledge that serves humanity. Thank you.
Thank you, Dr. Arun. So I take this opportunity to inform everyone about the India International Science Festival, IISF 2024, which will be held at IIT